Our second speaker is Ulrich Elling. He comes to us from Vienna, from the Institute of Molecular Biotechnology. Uli studied in Regensburg, Germany, and got a PhD from the IMBA in Heidelberg. And then for his postdoc, went to Josef Penninger in Vienna and has done some fascinating work on haploid cells. And this is what he's going to talk to us about today. Thanks very much for joining.
Yeah, thank you very much for your interest in the talk. This is going to be another academic talk, so bear with me. On this slide, you can see sort of a representation, what it looks like inside our cells. It's a big mess when you look at it. It's, of course, a great order also. Cells consist of a wide variety of different macromolecules and so on, metabolites, etc. And so if and these cells are capable of taking all kinds of inputs and processing them towards all kinds of outputs and responses, of course. And if we want to interfere with them, we have to, of course, account for that complexity within the cell. And one way to do this, as we've heard during this meeting as well, is phenotypic screening. Another way is, of course, if you want to do target-based screening, to identify the most relevant target within the cell. And this is what I'm trying to show you. I tried to show you a sort of novel approach for that. And because of the fact that all these molecules are very different in their biochemical properties, we sort of have to find one way to deal with them all, or at least as many as possible. And because they're all either directly in case of proteins or indirectly in case of metabolites, for example, encoded in our genome, a very simplistic way to deal with them is to simply do genetics. So look at molecules on a genetic basis. And the ways to do genetics are classically random mutagenesis as well as gene targeting in mice, for example, as we've just heard in some beautiful mouse experiments. But this is usually leads to heterozygous phenotypes in cells. And so it always requires breeding steps and is quite laborsome. Therefore, the big revolution was when RNA-based approaches, siRNAs, hRNA came on the market, where one could do knockdowns directly in the cells of interest, and that was much faster. However, these knockdowns are then either only transient in case of siRNAs. They're also not always very efficient, and they usually have off-targets. Everybody talks about genome editing nowadays, zinc fingers, TALEN, and now, of course, CRISPR. Also, these systems, of course, have off-target, and like other RNA-based approaches, have scale and efficiency issues. Also here, homozygosity is not always given. So in an ideal world, we would really want to combine all of these advantages from the different systems to make some sort of a conditional high-throughput homozygous approach possible. And there's one simple reason why this is not possible in mammalian cells usually, and that is diploidy. So, of course, as we all know, mammalian cells have 2 chromosomes each. And so whenever we introduce a mutation on one chromosome, there's a wild-type allele that can cover up for it. And that situation is very different in haploid organisms such as yeast. And so we can, of course, do very efficient screening approaches in yeast that are not really possible in mammalian cells. There is a few mammalian cells, and namely germ cells, that are, of course, haploid. And I try to make use of them to derive cells that would function somewhat like mammalian yeast. So the way to do this is to take mouse oocytes, and then one can activate them to start early embryonic development by exposure to 5% ethanol or strontium chloride. We brought them back into recipient females and then grew blastocysts out of them. However, this approach had been used a long time ago, actually, in the hope to derive haploid cells. And all the ES cells that were derived via blastocyst outgrowth from such blastocysts, such parthenogenic blastocysts, they were always diploid when looking at the chromosome content. My theory then was that maybe some haploid cells, shown in orange here, would still remain within these ES cell cultures, since they have been derived from a haploid oocyte, and that simply during the process of outgrowth of these ES cell colonies, such cells would be lost. And so I extended the old and well-known protocol simply by FACS sorting in the hope to identify a subpopulation of haploid cells. And after several rounds of FACS sorts into the dark matter, really, I could indeed identify a population that was strongly growing and that had a 1N and 2N DNA content indicative of a haploid genome. We confirmed that doing chromosome spreads, and as you can see here in 2 independent cell lines, we see exactly 20 chromosomes as you would expect from a haploid mouse cell. Furthermore, we wanted to look whether the cells that we had generated here were ES cells, and one way to do that is to look at ES cell markers, so OCT4, SOX2, NANOG, shown here. Both of the cell lines I analyzed were very positive for these markers, so you can see that these ES cell clusters, it's by the way 3D cultures, so the ES cells grow in three-dimensional colonies of up to 10,000 cells or so, a colony, on a feeder matrix in this case, but meanwhile also feeder-free. So they are positive for all these markers, and they are indeed ES cells. The other important feature of ES cells is, of course, the capability of differentiation, and the cells we had derived were agouti positive, so contributing to a brown fur. And when injected into a black 6 blastocyst, they contributed to coat color and also other organs within the mouse, so we could show that the haploid ES cells were indeed ES cells. This is one more slide I want to show, just to give you an idea. So both ES cell lines, like a control, can really, in a teratoma assay here, give rise to basically all germ layers of the mouse. So these cells are, despite the fact that they're only haploid, capable of differentiation in vivo, as shown here, but also in several in vitro assays. Well, it was unexpected that haploid cells could be viable, but that was not the reason why we did it. The reason why we did it, as I alluded in the introduction, was because we wanted to do more efficient genetics. And for doing so, we used splice acceptors that we would introduce randomly into genes, and that would then trap the transcript and thereby destroy gene function. We used the so-called double flex system, an array of inverted LOX BNF-RT sites that allow for the inversion of this splice acceptor, and thereby inversion would lead to, so splice acceptors would not be recognized anymore, and the gene function would be reverted back to wild type. Additionally, we modify our vectors in a way that we introduce internal barcodes into all of them, so we can identify mutations by an internal DNA tag. The nice thing about this system is that by inserting DNA, and that's the big advantage of a chemical mutagenesis, for example, we have a piece of DNA available, so we can, with an inverse PCR strategy, amplify adjacent DNA next to the insertion site, and thereby identify immediately the type of mutation we have introduced, and that can then be validated by a gene locus-specific PCR, as shown here in this panel of 9 mutations. More importantly, and because we have haploid cells, indeed, then of course we can also show that the wild-type locus is depleted. So indeed, this is, allows for recessive genetics then. I would like to stress that this is a very tight way of doing genetics, because we are starting with one cell line that we usually subclone beforehand. Then we mutate these cells all in parallel at the same time. And because of that possibility of inverting the splice acceptor, we can basically subtract any clonal effects from our mutants by reverting each of these clones that we get back to its wild-type sibling. So we can really do a very controlled way of genetics in a genome-wide scale. And that's important to reduce the noise when working with such high numbers. The way that we introduce our mutations is by making use of retroviruses, lentiviruses, or various types of transposons like TOL2, Sleeping Beauty, or Piggyback. Each of these systems has its own hotspots, and without carrying you through all the details here, but if one combines, for example, retrovirus, lentivirus, and TOL2 transposons, one starts to saturate the genome much better. Each column here would be one gene, and then this is showing the number of independent integrations within a gene within 1 million integration sites. Gene expression is shown here, so of course you have a tendency to express genes to be targeted, and that's of course good when doing mutagenesis in this case. In principle, we can do different things with such high cell numbers that are all individually mutated in one particular locus. And so I'll first talk a little bit about reverse genetics and then come to the forward genetics side. So for reverse genetics, we can of course use these pools of mutated cells, typically 50 million or so independent cells with each carrying a different mutation. And we can single them out and array them. And we do that using a robot-assisted cell culture system that we established, and then also four-dimensional pooling of DNA. And when doing so, we can basically identify the mutations of 10,000 clones at once. So we will then know for each of the 10,000 clones that we have in our freezer what mutation it carries. And this is the current standing of where we are. So we have picked around about 100,000 cell lines at the moment. 46,000 of them are already identified for the mutation. It hits, so we have in 20,000 of them we have mutations within genes. And we are covering at the moment 11,000 different genes. If you only look at clones that have a single mutation that we have introduced, we're covering about 8,000 genes. So we are really getting to a number where we can look at a substantial number of the genome directly by having the cells available in our freezer. These cells can then, of course, be rearrayed depending on certain assays that we are interested in and tested there. I'll just show you one example. It's not necessary to give you examples for what genetics can do, but this is, for example, in vitro differentiation of these ES cells into endothelium. And it is well known that Notch1 plays a role here. So if we just take a control clone or various clones mutated in Notch1, and then we add CRE or do not add CRE, then we can see that if the integration is in sense beforehand, then we have, of course, mutated the Notch1 locus. But if we then add CRE and turn it to antisense, so we turn the splice acceptor opposite to the transcriptional direction, we recover the Notch1 protein. The same in the opposite direction is true when the integration is antisense beforehand. And then if we do endothelium formation, we can see that loss of Notch1 strongly induces endothelium formation. That is well known. And we are now establishing sort of low throughput screens to go through candidates of endothelium. This archive of mutated ES cells will be made public to the scientific community, and we are already collaborating with a number of researchers on it. And I really want to emphasize that this effort would not have been possible without the help and collaboration of two companies that we are in close contact with, Nestle and Novartis. For the second part of my talk, I would like to come back to the target identification part, namely showing you the power of haploid genetics for doing forward genetic screens. And the way this is done is basically starting with a pool of mutated ES cells, carrying them through some sort of a selection in batch, so all the cells together, and identifying all the hits using next generation sequencing technology. This pipeline we have allows for identifying several million mutations within a single lane of deep sequencing, so it's very efficient to do this in high throughput. One of the examples that I can show here is Artemisinin. It's used as a treatment for malaria, but also cancer. And it's one of the most prescribed drugs in the world. It's got more than 100 million treatments a year. The mode of action is not entirely clear. We did this in collaboration with Novartis, who had told us that yeast minute mutants, so the mutants lacking mitochondria, were resistant. So it was clear that somehow the action of artemisinin had to do with the mitochondria, but it was not entirely clear how. So what we did then was a screen in ES cells. And we've seen all kinds of very fancy screens over the last 2 days. I just want to emphasize again that I'm not very fancy. This is what a genome-wide screen looks like for me. I just have a few dishes that I feed for typically maybe 3 weeks or so. And then in the case of artemisinin, I used a lethal dose of artemisinin, so only those cells that would survive that lethal dose would survive in here. They would grow out as colonies. I would pool all of these colonies, extract DNA, run the PCR protocol, put it on the deep sequence, and identify the mutations. And then I just look at which of these genes, which genes have many, many independent mutations, up to way over 100 independent mutations, indicative of a relevance in the pathway. And when I did that for artemisinin, I basically found a single biochemical pathway, porphyrin biosynthesis, and I put the p-values of the enzymes that I identified within the pathway above the respective enzymes. So you can see that I retrieve every single enzyme between 5-ALA and heme with p-values between 10 to the minus 15 and 10 to the minus 200. So it's more than just suggestive that this pathway is playing a role. And that's typically what one sees with haploid screens. It's not a sort of a list of putative targets. It's really the identification of targets. There's not really a gray zone in almost all screens that I do. Nevertheless, of course, one would like to validate the finding. In case of artemisinin, that was maybe almost over the top, but I did it anyways. And the system allows for that because we have a virus that carries either GFP or cherry. And the cherry then also carries a CRE, and because the CRE will invert the splice acceptor, the mutation that is putatively a knockout before, if playing a role in the pathway, should then be reverted back to antisense. So if I just infect if I pick 96 clones from my screen and array them in a 96-well format and infect them with those viruses, and then in a high throughput sampler for TESA, FAX approach, basically just compare the population of originally mutant cells with now putatively wild-type cells, then I can immediately validate the finding. And what one would expect from a control cell line is that independent of exposure to the drug or not, the ratio of the red and the green cells, so the mutants and the revertants, should be around 1. But in case that I have identified a relevant gene, then there should be a selective advantage upon drug exposure for the mutant cells. And that's exactly the case. So if you look at just a single 96-well plate here, you would expect the background is at 1, and you can see immediately that many clones are spiking out as having a dramatic selective advantage in the mutant version. And if we then focus on them, and map the mutation within these clones, immediately it becomes obvious that there are many independent mutations in the same genes again. We map it back to the path when we can say that we immediately validate these genes. So this is not only a way to identify genes, but it's also a way to immediately then validate them. And the nice thing about it is that you then also have clones in hand. So you have biological material that you can further work on in your assay. So what we can conclude from this is that artemisinin is toxic only in the presence of porphyrin, so heme, and that, of course, also makes sense if you think about the biology, because artemisinin actually kills malaria-inducing plasmodium, as I said before, and it does so only in the red blood cells, where there is a lot of heme. So we were quite happy with that result. But I had mentioned before that it's also but artemisinin is also being used in cancer, and as I've just shown to you, it's also lethal for ES cells at certain doses. I then looked at various cell lines of breast cancer and pancreatic cancer and so on, and you can see that in high concentrations, artemisinin is actually toxic for cells, and that is exactly what's being used in cancer treatment then. Now, because porphyrin is found all over in our body, but of course in the red blood cells that are not biochemically active anymore, it's not freshly produced regularly, we were thinking that if it was possible to deplete porphyrin from the body, we should actually be able to alleviate side effects induced by artemisinin treatment. So if we would inhibit porphyrin synthesis, it should actually deplete or reduce porphyrin levels in the body everywhere but the red blood. And we tested an inhibitor that Novartis was nice enough to supply. And as you can see here in ES cells, even at very low concentrations of the inhibitor, we could rescue cells from artemisinin-induced lethality. The opposite is true when we want to treat cancer. ALA is actually used in the clinic to identify certain types of tumors because they take up high amounts of ALA and thereby become autofluorescent, and so surgeons use it to identify the tumors. So we were thinking if we could load up tumor cells with ALA, whether they would then become more susceptible also to artemisinin treatment, and that's what we tested then. By addition of ALA to the cell culture, and you can see that in all cell lines tested here, the sensitivity to artemisinin actually increased in this experiment. Now, that's well and fine, but that's, of course, a lethality-based screen. So if we really want to look at a wide variety of different things, we would, of course, also be interested in doing other types of screens, and I'll show you two examples here. One is looking at de-repression of silenced elements. Our genome consists of 40% mobile elements that are silenced, and we wanted to understand how these elements are being kept in check and silenced. So we introduced a puromycin GFP cassette into the genome and identified clones where this cassette was integrated in align or LTR elements and thereby silenced. The other experiment was looking at one particular mobile element which actually reports for totipotency, so even more potent, if you wish, than ES cells, because ES cells are only pluripotent, they can only give rise to embryonic structure. 2-cell stage can still give rise even to extra embryonic cells. And 1% of ES cells in culture are actually positive for this reporter, so we used it to introduce it into our haploid ES cells. And then basically with the same strategy, we're planning to look for clones that were growing out and being puro-resistant. So we generated those reporter cell lines in the L line and LTR and 2 independent reporter cell lines for this particular endogenous element, mutagenized it, and then essentially took it under puromycin selection and identified the mutations. And again, we only found a very small number of cells and with a number of genes, excuse me. And the interesting thing was that then the overlap between the 2 independent screens was always 100%. There was no overlap between those 2 screens. So it was a very, we were very confident that the genes that we had identified actually made sense. In the de-repression, we found only genes that are vaguely involved, probably in epigenetics, but nothing really is known about these genes. They're all novel genes. For totipotency, we found transcription factors that are known to play a role in early embryonic development, but not much more. We then went ahead and validated them using shRNAs and to our great pleasure, they all validated also with shRNAs. We're currently working on them. I can show you some follow-up studies. So if we do RNA-seq on the de-repression, we actually see upregulation of certain types of satellite repeats, for example, in knockout of these genes. And here's is just a screenshot of the genome browser, and you can see that in the knockout situation, certain satellite repeats are massively expressed while they are kept in check in the antisense orientation of the integration. We knew, of course, that these mobile elements are being discussed in cancer, so we wanted to check what the role was in cancer. And we looked at the AML model, mouse AML model, and could see that immediately upon introduction of the hairpin, within a few days of expression of the hairpin, 5 days, these cells all showed signs of differentiation. You can also see it here in a spin assay. In all cases, not in the controls, but in all cases of these genes, we get, to our surprise, and we don't understand that yet, a full differentiation of these tumor cells. And also when we do a competition assay, we can see that basically within a few days, the cells that have depleted these genes drop out of a co-culture with wild-type controls in a similar kinetic than knockdown of a MYC would do. Yeah, this is also something we're following up. So I hope I could convince you that these haploid screens are quite a pleasure to do because they're very fast, they're saturating, they're highly specific, and they really can identify novel genes within a very short time, because really all you do is you expose your pool of cells for a few weeks to your treatment, and you can even use an internal reporter and couple that then again to lethality by using resistance like puromycin. But of course, depending on the assay that you want to run or on the selection you want to run, positive selection is not always the way. If we do positive selection, I really would be happy to admit that shRNAs or CRISPR screens will also work, because even if not all of the cells within a population, so the ones with red border would be intended to be hit with a, in gene X, then some cells it will work. So the hairpin will work in some cases, CRISPR will work in some cases. We select for those, they will do positive selection. We end up with these cultures and we can look at them. That's the case for many screens and also for the screens that I've shown you so far. But the situation is very different when we want to do negative selection, and that's much more complicated, because there we have to reach that horrible level of 100% efficiency. So if we target a gene X with an shRNA, there will always be cells that harbor the shRNA, but they don't do efficient knockdown, and they will then not deplete in a negative selection. The same is true for CRISPR. There will always be a few cells that don't do homozygous depletion of the locus, or only in-frame mutations, and the number will be substantially high. But even if it's only 10% of the cells that are carrying the agent that you introduced, then the best possible negative selection you can reach is from 100% presence here down to 10% presence, and that is only a 10-fold depletion, which is something that in pool is very, very difficult to measure. And we believe that the situation is very different when working with haploid cells, because in haploid cells you have independent mutations in each of these cells, and if you now apply negative selection, some of these mutations might only be hypomorphs and they might remain, but other ones will be loss of functions and they should then deplete. And because we actually look at each of these events independently, because they're all independent genomic mutations, we will see all of the ones that do true loss of function to completely deplete, and that is then a depletion that should be quite straightforward to identify also in batch experiments. So we believe that in particular when working with depletion screens such as synthetic lethality with cancer or drug target identification or drug sensitization, etc., haploid cells will be the way to go. So in summary, I showed you that we have generated haploid ES cells and improved on the saturating mutagenesis using a wide variety of different agents to introduce our splice acceptors. We have generated a gene knockout resource now in ES cells called Haplobank in collaboration with Novartis and Nestle, as I mentioned. And we are doing rapid saturated genetic screens, both lethality as well as reporter-based. With that, let me thank a lot of people. Joseph was my boss where I did my postdoc. I'm collaborating a lot with a friend and collaborator, Alex Stark. Some people from his lab, also Julius, both of them I actually know from my PhD time at IMBA. Johannes Zuber is the guy doing the hairpins with me. The people from Haplobank generating that archive, and Dominik Höpfner and many, many others from Novartis for their collaboration as well. Thank you very much for your attention.
Thank you, Uli. Very exciting, very powerful tool. Questions from the floor? Stefan?
There's a haploinsufficiency, monoallelic expression, and all kinds of other phenomena which would suggest that haploid cells are not identical to diploid cells. To what degree is that true if you look at functional parameters, and especially if you would look at more differentiated cells?
Thank you for that question. Yeah, many genes are known to be haploinsufficient. So if the gene is there only heterozygously in a diploid context, then the cell is lethal even. It's very hard to rule out that there is an effect of haploidy to the cells. If you think, for example, about let me start the other way around. So my ignorant view is that in the end, in a haploid cell, most things are very much the same again. So you would have only one locus of a transcription factor that thereby only transcribes less of that transcription factor, but there's also only half the volume of the nucleus where the proteins of that transcription factor have to, so the gene products, have to float around. They also have to only bind to half of the target sites within the nucleus. So in the end, most things stay the same because haploid cells are only half the size of a diploid cell. That is a bit of a simplistic view, I have to admit. There's exceptions to that. If you think, for example, about volume to surface ratios, they should change when cell size changes, and we haven't addressed that yet conclusively. Another issue is X inactivation, and I didn't go into that. So ES cells are one of the few cells that actually have both X chromosomes in female case active, and differentiated cells will inactivate one X chromosome. So the ratio of X to autosome is usually 1 to 2. In haploid cells, you have one X chromosome and one autosome, so the ratio is 1 to 1, which is only tolerated in ES cells. And like I say, usually X chromosomes will then inactivate during differentiation. If we differentiate haploid ES cells, we see that they spontaneously become diploid. So even within ES cells, they do become diploid over time. So every now and then we FACS sort them again to keep them haploid. But if you differentiate them, they really become diploid quite effectively. And differentiated cells are always diploid. I believe that it's simply due to the fact that both the double dose of X as well as no X is lethal.
One more question? If not, thank you very much. Everybody's probably eager to load up on caffeine for the morning, so enjoy the coffee break, and we'll be back at 11 o'clock.